part of thesis, they are forced to do it. Any message for students as how they can do quality research, just not publish something? And can a clinician in a clinic also do research?
So, yeah, Dr. Manisha, it's a wonderful question. And actually, as a basic scientist, my lab is mostly an immunology lab. And we try to, I have a lot of collaborators who are clinicians. And I just wanted to share that we have published Nature and Science Signaling. All this publication has come from the work with the clinical work made the foundations. So coming to the first question, what I want to say is how the choosing the research topic for your thesis work. So it's a very general question, and I think, and it's very important, like what to choose the topic. So first thing is that topic should be very interesting. It should touch your heart. And that the single dot, it should connect to your career. So you can have a multiple thesis topic and the novelty is there. And it should be able to translate from bedside to bedside. So I say that because you're a clinician, from bedside to bedside and back to bedside. So that's the circle. And again, you should be very novel, translatable, and it should be has a potential to commercialize, because now the funding agencies, they are mostly focusing on like what is that translational capability of any research, and then it's novelty part, and you should focus more on before doing anything, the focus should be on your research review of literature. If you don't do a proper review of literature and find that, okay, something is existing, so you missed out the novelty part. So can I combine with the second question also? Yeah, and absolutely the research, the groundbreaking research is coming from the clinics. I will give you two examples where, as I told you, we were able to publish in, one is a Nature and another is a Science. So how we were published, because both the things, the basic foundation came from the clinics, where we found that the first study was about how some of the successful vaccine works. And this was the basic question we took. And then we started working with the clinicians, and we took the volunteers, immunized with the vaccines, and we found that and we took the model, yellow fever as a vaccine, which is one of the most successful vaccines. And when we started working through AIML, we did a lot of transcriptomics, proteomics, and all systems immunoinformatics. We found that one of the genes which nobody has reported was popping out in the clinical samples. And when we started working with the mechanism, it was found to be a very old, the pathways was not known. We started working with the mechanism, and ultimately what we found that this gene was responsible for programming of vaccine-induced protective immunity. And with this knowledge, we wanted to translate into clinics. So how we did, we make a small molecule against this, whatever the gene we identified, and then we went to our companies, and they were very helpful in making and designing the novel activators for this. And when we applied, this whole finding was applied for developing a first antidote, which is an engineered antibody against COVID-19. And I'm saying this is it completed human phase 2 trial, but by the time we went to a phase 3 trial, we are not, the COVID went. So it's still in the process. So that whole platform we are using for other, making antidote for other therapies, the infectious diseases. So this is one of the classical examples that the basic foundation was from the clinics. Another example I will give you is that we have started working with the psoriasis. And see, there is a lot of problem, I will tell. There is the clinics and academician, lot of amalgamation problem is happening. For getting 11 patients, I had to struggle 3 years. Okay, and somehow using that whole AIML and immunoinformatics approaches, again we identified one of the genes that was highly upregulated in the psoriatic patients, and we validated, we collaborated with the Apollo Hospital. They were generous in giving the biopsies. Somehow we were able to identify that, validate the target, and that it is the first time molecule that is found to be important in programming causing psoriasis. And we collaborated with Dr. Reddy's. Dr. Reddy's is generous to fund it, and we have designed an inhibitor against that. And this is the first type of inhibitor which is not against any cytokine therapy. It is a skin-specific gene that is upregulated in the patient sample. So what I mean to say, many of the information is coming from the clinics, and as a basic scientist, a lot of mechanistic studies, and try to provide a solution to it. So it has a huge potential where clinicians and academicians can come together, and I think it could be we can do a lot of magical science together.
Thank you for that excellent answer. So what we understood is, see, don't just try to copy anywhere from internet, do what is troubling you, the question which troubles you. And I'll give you an example. This is an idea clinics, lot of diabetic patients you see. So this was, many of them go for dialysis and transplant. So how do we diagnose diabetic kidney disease? Traditionally by checking the urine protein, and we also check the creatinine. These are the tests everybody here is familiar with, that all diabetics, we do this and check whether they'll develop kidney failure or not. But both these, creatinine and urine protein, are late markers. By the time we identify, the patient already has severe kidney damage. So we thought, Rakesh sees a lot of diabetic patients, and you all see, I'm sure, a lot of diabetic patients, and we see a lot of kidney failure patients. We thought that we did a literature research to find out, can we diagnose this kidney disease even earlier, before the protein and creatinine become abnormal. So for that, we found something of metabolomic tool, which can be tested in the blood samples. And what we did was, we collected samples from large number of people, those with kidney failure, without kidney failure, those with diabetes, those without diabetes. And we studied this metabolomic marker in all these patients. We couldn't do it ourselves. I work in a government hospital, and again, we don't have metabolomics at our disposal. So we did collaboration, which Dr. Nuruddin also highlighted. So we collaborated with IIT Bombay, and they provided us this tool free of cost. So once this idea we got, what we did was we wrote a grant proposal. This was approved because it was a novel idea, trying to find out whether we can diagnose kidney disease early. And in this, we found a biomarker. So now we have published the preliminary findings that were published in a high-impact journal, and now the second phase is to follow up these patients to validate, and we have involved other centers also. So single center study, now we are making it multi-centric to validate our findings. So this is again what was bothering us, any new marker for diabetic kidney failure, and that's why we started it. So the first point is, do something which is bothering you, not because something is published in high-impact journal. Publish something, work on something which is common in India. So I hope the message is clear from Dr. Nuruddin. Be passionate, not for show. Work for cause, not for applause. So now going to Dr. Aarti. So she is at BITS, Hyderabad, and her question is, ma'am, how many types of studies are there, and how do the PGs choose, and how should the clinicians choose what suits their research question?
Okay, so I agree with one of the points which you mentioned, that you have to look at the problem. Although in the literature you will read, there are lots of different kinds of studies. You have case studies, case reports, cross-sectional studies. I will not go into that because all of that information you will already have. These days we have a plethora of information available online. So when they told me to share the answers for that, I told them I will directly speak instead of writing it on the PowerPoint. I think before deciding on the various types of studies, as Dr. Sahikal correctly mentioned, it is very important to understand what is the problem. Because unless and until you don't know what is the problem, you cannot decide on different types of studies. So first and foremost, India is a very heterogeneous country, and we have our own set of health problems. So whenever you are going into the preclinical or clinical setting, the first and foremost thing is to identify the problem. Unless and until you are not into deep understanding of the problem, you are not very attentive of the problem, you will not be able to figure out the solutions for that. Because unless and until your reading, your literature research is not very thorough. So the solutions will not come out. So first and foremost, I will suggest both for clinicians as well as the preclinical students who are here, that whenever you are dealing with, specifically let us say clinical students are here, whenever they are dealing with any kind of patients, you have to be very, very attentive with the case history, and how frequently that particular event is occurring and why that event is occurring very frequently. This is very, very important to know, and then analyze this or observe this data very carefully, because your attention is very, very important to understand that particular problem. You have to stay with that information for a long period of time. So pay attention to what is the observation that is happening. Unless and until the attention is not there, what kind of, why this event is occurring very frequently, you will not be able to go deeper. So first and foremost, understanding the problem much more deeper, a thorough literature research is very, very important. And then at the clinical stage, you have case studies and case reports, where a single case observation which has been not reported before, you can record that and go a bit detail into that particular observation. Another important thing which you can do is that any kind of case you are observing, you see a very unique finding, you try to report it in the form of the case reports or case series. Now, another thing you have to pay attention is that whenever you are reporting any case series or case report, it should be very, very genuinely done, and it should be authentically done. Reason I'm just saying is, when you are entering this data into the data system, this data will be getting extrapolated, and scientists are going to read this kind of data. There will be a time when not much of the clinical trials will happen because the technology is changing very rapidly. AI is coming into picture everywhere. And there will be a time when your data that you entered into the system will be extrapolated for figuring out the treatment of a disease. So it is very, very important that you make those observations and publish those observations, irrespective of the impact factor. But those observations should be very, very thoroughly and keenly done. And when you enter it into the data system, data will extrapolate it and interpret it. So it is very, very important that you pay attention. Attention to the problem is very important. And then later on, it gets extrapolated. So clinical trials require significant amount of time and money. And technology is developing in such a way that later on, how to reduce the cost and how to reduce the time. At the same time, you have to look at the India also, which is a country where 70% of our population is not having access to the healthcare. So in that case, we have to make technology affordable. So taking those factors into consideration, whenever you are studying any research, you have to see what is the problem and how much that problem is bothering you and how much you are ready to go deeper into that particular problem. So then later comes the case studies, case reports, ecological studies or analytical studies. They will come much later. But before that, your thorough understanding of the problem is very, very important. And you can publish them in the form of short papers, but they will be entered into the data system. Remember that your data is very, very critical because it will be there in the system for throughout the life. So make sure the data is accurate. So that's
thank you. Thank you, Dr. Aarti, for making that point. Be honest about the data. And now, for example, clinicians, as Dr. Aarti said, the easiest study is the case report. So we really see some cases which are very unusual. Start with that. One single case report, but be honest. Then you have major type of studies which the students should know. So you have, for example, cross-sectional studies. Cross-sectional, when do we use? When we want to find out prevalence. Suppose you are sitting in a diabetic OP and you want to find out how many had heart disease. So you note that in your register. So that type of study can be done at one time point, and cross-sectional study. Then you have case control studies. So case control means, now for example, I am a nephrologist. I see a lot of patients with diabetic kidney failure. So I have, say, around 100 patients of diabetic kidney failure. I go back in the history and see whether which person had poor control, which was a smoker, which was an alcoholic, which had a family history, and try to find out among all these cases and the patients who do not have diabetic kidney failure, what was the difference? Whether these people smoked more or whether they drank more alcohol. So we are looking at the history. That's case control. Then what's a cohort study? So that's very good study that I start today and look into the future. So diabetic people, 100 people, now future I am seeing which are developing, which are not developing. So cohort studies, we look into the future. Then randomized controlled trials. So for a clinician and as for postgraduates also, this is the best form of research. Now what do you mean randomized controlled trial? So the name itself shows that we have randomized people. into one group and the second group, and everything else remains the same. But maybe for this example, in a diabetic patient, I am giving semaglutide injection in one arm, in the other group I am not giving. And we find out whether semaglutide is nephroprotective. Less people develop kidney failure when we give semaglutide. So this is a randomized controlled trial. As Dr. Aarti said, there are hundreds of other studies. But please remember these: case control, cohort, cross-sectional, and RCTs. Because these are the most common. And as clinicians, don't be so worried by so many number of studies. You can do any of them, but you need time. Start with reporting a case and start from tomorrow. So that's the message from Dr. Aarti's explanation. Now we have Dr. Rao, who will enlighten us about how to frame a research question. So you have identified a problem. So what elements should be there before we collect data on that? Dr. Rao.
Thank you, Manisha. First of all, I was surprised to see such acronyms, because we were never told all these things. I was mentioning to her, I said, what is this? Why do you have to talk about all such things? But she said, the current postgraduates are so much well equipped and knowledgeable with the research methodology, and she told me that even the teachers are also trying to be good into research methodology. It seems
Absolutely.
it's also part of the...
So PICOT and FINER are parts of our exam questions. And for youngsters, it's important to have acronym because that's how we mug up things, right? So PICOT, you'll remember all elements. FINER, you'll remember all elements. That's it. Over to you, Dr. Rao.
I know acronym is okay, but I'm like an old man, so in olden days, we never had such acronyms, but we have also done a lot of research.
Absolutely.
So I wanted to show you my own examples. Okay, PICOT and FINER, it is there everywhere. You can read and mug it up. Madam said there may be questions. People will, you may be asked about the questions in orals. Next slide. Okay, next slide, just one slide, one slide.
This one, next one?
Okay. Yes. Yeah, well, that stop it there. So long ago when research was not a great thing to do and everybody was against doing research, my parents, my friends, my family, my colleagues, my doctor, everybody was against research. After completing my MBBS, I started doing, I don't know why, just to make myself better than others, because I was not a good student. When she was introduced, she was told she had gold medals. In all courses, she had gold medals. I never had gold medals. But I wanted to be at least known differently among my circles. As a doctor, as an MBBS doctor, so I thought, so what else? I cannot excel in sports. I was not a good sportsman. I did not know music, I did not know song, I did not know anything. So I thought, why can't we do some sort of a research program and try to present it in conferences and make yourself known? Okay, that was the only motive. I was just thinking, why did I do it? That is the only motive, just to make yourself different and superior to others, because you cannot excel them in science, you cannot excel them in marks. So I went to... And first thing, please remember, all of you, if you want to do research, you have to have a guide, a godfather or something like that. If you want to do real research, see, I think you told me, for 11 psoriasis, they had to struggle for 4 years. And in a single day, you can have 100 psoriasis in Usmania Hospital. So it's not a big thing.
Collaborate.
But he said you used the word amalgamation. So if you want to do research for question, whether you want to FINER or PICOT or such things, you have to go to a institute like PHFI or university. University is a very big thing. They have so many people there. Or to this is a new institute that came to Hyderabad. I'm not familiar with them, but BITS Pilani is a very big name. So I met some undergraduate in the morning. He's from Mahavir Medical College. Is he here? Yeah. Is there? Who is that Mahavir Medical College undergraduate? I asked him, why are you asking so many tricky questions? He said, you remember in the morning somebody was asking all the complicated questions. Then he said he is going to CCMB. You know him? Yeah. He is going to CCMB weekly twice to learn about the metabolomics. And madam collaborated with IIT, Bombay. Like that, you should have some sort of a collaboration where your other people will help you. So I have collaborated. I mean, there was no collaboration, but there was something called Senior Research Fellow after MBBS. You can apply for SRF in ICMR, and then they give 3 years. Usually it takes 3 months. Even now also it takes 3 months. You will be paid almost like 40,000 rupees as SRF post MBBS. But it's a lot of paperwork. You have to do paperwork. So I have done the paperwork, and I worked on rabies. I took rabies because nobody works on rabies. If you do anything on rabies, people will recognize you. So I was the only single person in the entire city of Madras touching rabies patient. I was doing corneal smears. Like that, do something different. No? Do something different. So my thesis was anti-mortem diagnosis of immunofluorescence staining of corneal smears for anti-mortem diagnosis of human rabies. So like that, take something new. You can learn it. Now the internet is there. You learn it yourself, whatever is new. Whichever gives you recognition, that's important because you need recognition. Not to be a great scientist, but you want recognition among your own colleagues. Your own colleagues are always much better than you in answering questions to your bosses. Bosses are always impressed by your colleagues, but not by you. So impress your bosses, you have to do something else. That is my idea of finer and...
So thank you, excellently put. And I am very smart. I am intermediate age. So I have taken experience from Dr. P.V. Rao and I have fused it with the AI generation. Because I have to answer to them, answer to my students, many of them going to CCMB and asking tricky questions. And that's the reason, using Sir's age-old wisdom and my smartness, I have come up with these criteria. Because you have to have all these elements in your paper. Otherwise, Dr. P.V. Rao will be the reviewer and won't publish our research. So, PCOT stands for population, diabetic population in our paper. Intervention. So, we did the test, metabolomic test. You should have a comparator. So, for example, when we are giving semaglutide to one group, not giving to other group, so comparator is a placebo. Outcome, we were looking at kidney outcomes, kidney failure and time. So have a time, 1 year. All this should come in your research hypothesis. Or if you submit to any funding agency, as Dr. Rao has mentioned, you need your project to get approved. So easy to remember. All PCOT should be there, easy to remember. Otherwise, we'll get confused and miss one of them. And finer, again, it reiterates what Dr. Rao already said. It should be feasible. See, I'm sitting here, metabolomics, not in Osmania, so collaborate with IIT Bombay. So we made it feasible. Interesting. So everybody would like to know, can we diagnose kidney failure early? Novel. It was not done before. Ethical. We took all ethics approvals and all that. Patient's consent as well. And relevant. Is it relevant? Definitely. India is facing a storm of kidney failure, dialysis, transplant, money. It is definitely relevant. So remember, ladies and gentlemen, PCOT and finer. And Dr. Rao will agree to that as well. Okay, thank you. So learned a lot from your experience and excellent answers, Dr. Rao. And you have been our mentor. So, I mean, we have always learned from you and our learning. So Dr. Shukla.
Can I add?
Yes.
Yeah, just I wanted to tell. See, most of the academicians, universities, they are writing grant. There is a section, do you have a clinical research person or not? So we have to, any translation project we are submitting, a clinical clinician has to be there if it needs to be funded. Likewise, that's the importance of clinical work. So and if you want to write, you need to show the mechanism. So we have to have a person who is a basic scientist. So that's the requirement, and now funding agencies, if you're going for big grants, they are asking these things.
Absolutely. That's why we have a mix here on the panel. And funding agency Idea Clinics is always there. Okay. So how to do literature search? So I'll put this to Dr. Shukla. Tips for beginners and tips for people who are doing advanced work.
Yeah. Thank you, Dr. Sahai. First, why do we do a literature search? Why do we need a literature search? So the idea, first, if you want to develop understanding about something new, just now Madam said metabolomics versus the traditional diagnostics for renal failure, renal conditions. Okay. You want to understand, so you need literature search. What is there? What is already existing before you actually decide into, and this is very important for all the clinicians. Many of us, because of our busy schedule, rely on medical representatives to give us this information. I think we should learn the literature search, literature review, and develop our own concepts. Okay, so that is the basic use. The second is, you have a problem. Like ma'am said, she had a problem. Kidney failure people in diabetes are coming late. Or when they come actually with the creatinine or renal function test going down, the kidney is already substantially damaged. Now, how do I identify this? So that's the problem. Okay, so when you have a problem, you want to do research, you want to go further into it, then you again want to understand what others have done about it. Research is all about building on existing blocks. There is existing understanding and you have to build on those existing blocks. Okay, so the literature search is that mechanism. It's fundamental, building on the current understanding. Then you need to know, now you have the whole idea, the concept is there, you have identified the gap, you have a problem, but how do I do it? Okay, so literature search, when you learn, you know that the way the computer or the libraries, the search is organized, there is a particular way. So there are, what you are thinking is key concepts. What you will search are keywords and medical subject headings. Okay, keywords are keywords, every literature, every scientific paper you see, under the abstract, there will be keywords given. Old scientific papers which have been published some years back will also have mesh terms mentioned. So that is where you will get your initial keywords and mesh terms from. So start with few papers, start looking at the keywords and mesh term. But before that, I'm not covering what they have already covered. So PICO has to be developed, so your question has to be there, your finer assessment has to be there, and then you get into this. Okay, so you have a question, you identify your key concepts, then you go into keywords and the mesh terms, which is medical subject heading. Medical subject heading actually allow you to search for multiple terms under them. They are used by the National Library of Medicine. All the research papers are analyzed and mesh terms, and they are categorized under, so they are under a hierarchical order. So you use a higher level mesh term, you will identify all the papers. For example, I'll give you the same example. Now, what are you looking at? Diabetes. Now, diabetes has 120 mesh terms. Okay. The broadest is diabetes mellitus. So you use diabetes mellitus, it will cover all diabetes, diabetic patients, diabetes mellitus, type 1 diabetes, type 2 diabetes. There are so many things that will come under, but you use the mesh term diabetic mellitus, it will cover all the things. So this is how mesh terms help. But use combination of keywords and the mesh term. The reason, new papers will not have mesh terms. Papers which are published in the last 1 year may not have, because there is a process they get categorized in. All the libraries, all the, the sources, so PubMed, Medline, all these sources, databases. Yeah, databases, they categorize, they go through this, there are librarians, there are experts working, and they categorize them. So recent papers might not be categorized. So always use keywords if you are interested in the recent paper. Then each key concept or keyword, combine them to similar group and search them together. Don't search for the whole question in one go. Each key concept with their mesh word or keyword, so that's where you use the Boolean operator or this, okay, and search. Once you have identified all the key concepts, then you combine them. And that is how you narrow down. So what you want in a literature search is you want a comprehensive search, but also very specific. If you go very comprehensive, then reading all that literature, बुड्ढे हो जाओगे. ठीक है? So you want to get specific literature, but you want also comprehensive. You don't want to miss out something important. So that's where you have to combine or initially, and then and as a Boolean operator, which will then bring down to a comprehensive search. So these are the steps, and remember it is an iterative process. So my check is when I do a literature search, I have few important papers that I know about in that area. And if my search strategy is not able to pick any one of them properly, that means it is not fine-tuned. Then I go back and revisit and try to understand what mesh term, why it has not picked up, and then fine-tune it. Okay. I think I have covered, I'm simplified. My only thing is the difference between the beginners and the advanced people is don't hesitate. It is not a difficult thing. It is not rocket science. It is something which will come to you very logically. You have to just initiate. People get scared about it. Research, literature search, very difficult thing. I don't know how to do it. No, don't. Get into it. Start doing through. Anything that I said is illogical. It's very logical. So once you start doing it, you will develop your own understanding of it and you will fine-tune it. So that is my advice to the beginners. Don't hesitate. Use it in your daily. Don't just use it for research. Use it if you want to develop understanding of any new concept. Use a literature search. वो research में बाद में आएगा. Start with that. You will migrate to research subsequently. Okay. So, and then I have this infographic, the last infographic, which actually talks of how you summarize, how you. So the important thing in literature review, what people do is people copy paste. He said so. Et al. So and so et al. Said so. No, don't get into that. That's again for advanced. Synthesized. Try to develop understanding and synthesize. Sometimes you can combine 2 or 3 authors together and say what they are supporting, what they are contradicting. That's synthesis. It is not just about what they said, what they did not say. It is about bringing the knowledge together and synthesizing and summarizing it effectively. So I will stop here. I will add on subsequently. Thank you.
So thank you for a comprehensive answer. So I think he has told us the basics of literature review. And as he said, don't get scared. I mean, start somewhere. All of us are playing with smartphones and learning so many things daily. We are becoming smarter by the day. You can start anywhere. So see, this slide, you can just take a pic, because if you have not understood mesh terms, Boolean operator, go and Google them, easy. So initially when I started, so what we did, diabetes, so I put diabetes, so many articles came. I thought we should collect more articles. But as Dr. Shukla said, you cannot read everything. Be more specific. So type 2 diabetes mellitus. And I now wanted kidney disease also. So I used the Boolean operator and. If I use type 2 diabetes or kidney, entire search will come, but I'm interested in both together. So use Boolean operator and. So Boolean operator, go back and read, important. Mesh terms, you should use the correct terms. And databases are there, PubMed, Scopus, Embase, Web of Science, Cochrane, Clinical Trials, Glay Literature. These are all database sources. So this is very, very important. If you have not understood anything, go back, Google and read, but be familiar with the terms.
Just one addition. Use the not Boolean operator when you don't want something. For example, you want only adults, so not pediatrics. You want only human beings, not animal. Okay, so that way, because research is a big field. When you start searching, you will find thousands and thousands of articles. So learn when to use not also, but that you will gradually get to it.
Yes. So thank you for clarifying that as well. So Dr. Rakesh, can you give some examples of good research topic for students and what could be the research topics for clinicians? I know that's a very broad question, but presuming they are all clinicians working with sugar patients, what do you think would be common points?
I think everybody has been giving very good examples and has given us an understanding of how do you identify a research question. Something which you're not understanding, something which is bothering you in your practice is a good research question. So in fact, today morning we were discussing about how NAFLD progresses, fatty liver progresses. So my question was, does it differ in Indians as compared to other populations? Because Dr. Sharath told us that it progresses very slowly. So my thought was whether it is the same in Indians. So that could be a good research question. We look at follow up patients with fatty liver disease and diabetes, and then you follow them up every year. You look at it within a year itself. You can get some answers, and that can be a question that you can go on with for many years also, because you can then understand how it is progressing and what is happening if it is not progressing. How is it going to remain stable? Is it going to reverse? So I think many things will be answered by that. So I think such questions should be kept in mind. I mean, if you're worried about, as Dr. Manisha said, about the kidney disease, whether you can pick it up early is a good question, because that is what you're seeing. You're seeing patients coming, and you don't expect that creatinine is not rising as you expected. It is happening very fast in some people, happening slower in some other people. So that can be a question that you are trying to understand. So whatever comes to your mind based on the patients that you're seeing, the people that you're looking at, and depending on what is not explainable by the available literature, or when you read about something and you find that there's something which is not explainable, that becomes a good research question. So I think that is what we should look at, because then it satisfies all the criteria. We said you should have you design a question based on the PICOT, but finer criteria is to see whether it is feasible or not. I mean, what you're saying is whether you can do it, because you're having patients having this problem. So you're trying to solve that question that is coming to your mind. And you have the population available, you then use appropriate tools to study that, and then you come up with a explanation for that. So that's how you should look at simple problems. Can be simple things which are not explainable by the available literature, can be good research questions.
Yeah, thank you for that nice answer. So what we have understood is you need not have a fancy lab attached to your clinic. Clinical problems are also good research problems. That could be a research question, right? So thank you for emphasizing that. Now we'll have a second round of the questions. That's the final round, and we'll have quick answers. So probably one minute answer from everyone, so that we can have some audience answers as well, or questions at the end. So role of biostatistics, Dr. Nuruddin. Statistics means bapre, hell. Yeah.
Yeah. So it's a very valid question when you are doing clinical research. Okay. So statistics is determine the robustness of your findings, reliability of your data, reproducibility. I'm not a biostatistician, and there comes the collaboration. So that's how collaboration is important. I have to collaborate with a biostatistician, and now not only interdisciplinary collaborations, we have a transdisciplinary collaborations, we have a collaboration with the anthropology people, we have a collaboration with the mathematics. So there is a concept called mathematical immunology. So that's how what we call is collaboration is very important. And coming to the question is, statistics plays very important. We have to determine the sample size, we have to write what type of statistics we are going to use. Because in clinical data, there is a lot of variability. Even there are a lot of outliers are there. So how we can remove outliers? So outliers, we cannot remove like that. So there are some statistical tests are there to remove outliers. There are tests which you want to use, whether you want to go for Mann-Whitney, whether we have to go t-test, whether we have to go other parametric tests are there. So those kind of things is required. And therefore, we should consult a good statistician. We should have a good collaborator.
Thank you. So one point I would like to add here for the students, as a teacher I realize, when do the students go to the statistician? At the end, when they have finished everything, and they tell the statistician, sir, we want something significant, some significant p-value, this is my result, please come with some positive result. That is the most foolish thing to do. Involve the statistician from the beginning. So that's important. And many of the students already know the basics of biostatistics. Now it's mandatory. BCBR for faculty also. So little bit at least, what do you mean by mean, mode, median?
Just one point I want to emphasize here is that statistics, one of the reason is that you have to generalize your findings to the target population. And when you are selecting the sample size, you select the sample size at a particular location. But when a medicine is introduced into the market, it is exposed to everybody. It is not only restricted to a particular region. So it will be like in Hyderabad, it will be in Delhi, it will be in Mumbai, but your patient population which you selected for your study was from a particular location, although you have multicentric studies as well. So for generalizability, it is very, very important that statistical analysis needs to be done appropriately. Extrapolation of a data and generalizability, that is the main reason. That's why statistics becomes very, very important, and your sample size is also very important for that.
Thank you for the input. So now,
yes. Add one thing for statistics.
Yeah.
By just using ChatGPT or Gemini, or I use the Chinese version, DeepSeek, we can make a beautiful photograph, paragraph of sample selection. You have any question in your mind, and you put it there and ask it to give you a statistical paragraph on sample size selection, on your how do you want to do your methodology, and within fraction of a second it is giving. So that is also a good thing to do.
Absolutely. And the other thing is, be honest. Don't cook up data. That should never be done. Dr. Rao said that he is not in touch with the newer technologies. I don't agree. He mentioned Gemini and he mentioned the DeepSeek. So no, your lie is caught, Dr. Rao. Okay. So now the next thing is how to apply for grants.
Okay, apply for grants. I have one slide here.
Sir, just a point on the research and the methodology. So don't worry about negative results. Be sincere, true to the methodology. If your methodology is good, positive, negative, all results are important. Okay, but methodology should be good and sample size should be strong enough to communicate your findings. Then positive, negative result, both are equally important. Unfortunately, we give more importance to positive result, but both are equally important.
Absolutely.
So some of the big groundbreaking discoveries are negative results. Okay, I will tell you the T-cell kinetics, if you have seen, that data is from the negative result. Okay, so don't worry about negative results. Okay, so
and publish them. Go for publication. Don't just hold back.
Yeah, this is a very common problem with students. So we had an ACHIEVE study, 3-year study, spironolactone in dialysis patients. It was a negative study. It got published in one of the major reputed journals. That spironolactone is not effective for heart failure reduction in dialysis patients. So we were so upset with the result. We were also part of the study. But again, it's an important finding, life-changing for the patients. Be honest towards research.
Yeah, just to add to that. So I think, this has been a phenomenon that has been happening, that people don't publish negative results if they have, made a hypothesis that, okay, this is likely to happen and that doesn't happen. then they don't publish it. So to overcome that, in fact, now what is required is that everybody is required to register your
Clinical
work
trial, yes.
on the what do you call as the
CTRI.
CTRI website. Once you put that, your methodology, your what you're planning to do, all that has to be put there to get a CTRI registration, and all these you have to upload your results also. And only when you have that, then that is one of the mandatory requirements for publication also. So that is again designed with the idea that many of the negative results don't get published. So you can look at that, and whenever you're searching also, you can look at the CTRI website to identify, understand what others are doing. Somebody who's working on a similar problem also, you can find that out by going to the CTRI website. I think Dr. Shukla wants to add something.
No, I'll not use the mic. This is very dangerous in the world of AI, not having negative results.
Yes.
Because it is queuing the data to one side, and AI will inform you only based on the data.
So be honest, because you are playing with patients' lives many of the times. So now we come to the crux of the whole thing. How do we do research? We don't have grants. So Dr. Rao is going to tell us how to apply for grants.
Next slide. So I have given a slide here, and which shows how I made money in the initial days on my own without going to the institutes. So I started with, I did not know anything about ICMR. I saw a newspaper cutting somewhere in 1978 that ICMR funds go a-begging. Then I found nobody told me, my professors did not tell me, but I applied to ICMR and they had replied to me. 3 months I got SRF. SRF was 750 rupees first year, 875, 950. So 3 years I had done SRF, and that's how I started getting into ICMR. And then once you get into ICMR or DST or DBT or any organization, somehow CSIR, you are assured of continuation of funding. That is the first thing. You have to somehow get into either CSIR, ICMR. All MBBS medical graduates are eligible for SRF without any exams. Non-medical people have to go for exams for JRF and SRF. So please think about that first. DBT as opening, CSIR as opening. I also worked as a scientific officer. That's called pool officer after MD. So that's also by application. It takes hardly 2-3 months and you will get money. Nowadays money is also plenty. That is much more than your central government employees. So that is one source of money what you get for funding. And please remember, I want to tell you most importantly that the money spent by India on research combined, CS, I'm talking about medical research, CSIR, DBT, DST, and all these things is proportionately more than what United States is spending. PHFI is a very large organization. That is one thing. Second thing is, which I have not done, those days PHFI was not there. Universities were not active. So those days, but now as young people, you have access to all these great institutions. You go to PHFI, and talk to them 3 times, 4 times, 5 times, 10 times, and they will do something about your funding. Go to university, School of Health Sciences or your university, they will also do. There is money there, plenty, but the only thing is you have to be in touch with them. And they will also be, this is mutually beneficial. You will be giving them clinical samples, they will be helping you with the funding. Then I think BITS Pilani is also IIT, all of them are looking for people to come and ask for money. That's one thing. Then I'm also a chairman of a research committee for Asian Healthcare Foundation, AAG, for last 10-15 years. AAG gives, but for AAG employees, not for others. Okay? So you be a AAG, you will get some money for your research. You are an employee of KIMS Hospital, you will get money from KIMS Hospital. KIMS Hospital has got a research foundation. Dr. Bhushan was there now. Dr. Manimala Rao, my madam is in charge now for research. Like that, nowadays, it's not like olden days. Those days, there was not much opening. But today, there's plenty of money everywhere. PHFI, then CCDC, what is this? Center for Chronic Disease Control. That is also giving money. So like that, now there are a lot of openings. Next slide. What is the next slide? I'm just showing, okay, this is fine. If you are a member of Nizam Institute of Medical Sciences, we have sources. All India Institute, we have sources. I'm just showing you these things. but you should be attached to some institute temporarily. I was never a permanent employee of All India Institute, but I was getting a lot of grants, including WHO grants. So like that, you have to be attached to an institute or to a person who will help you in getting the funding. There are plenty of money for research in India. India is one of the richest countries. India is spending more than United States proportionately on medical research. Please be sure about that. So that's what I want to advise.
Thank you. So Dr. Rao exemplifies where there is a will, there is always, always a way. So very nice, sir. So as he pointed out, now the concept given in India is made in India, make in India, and government is investing hugely in research. But we have to be smart. So you should go to these websites. You have ICMR website, DBT Welcome Trust, so many are there. So you should know which are the hot things. So tuberculosis is hot, NCD is epic. So again, I mean, that will give you idea where to focus your research on. So I think that's very, very important. You should write a proper proposal. Now suppose if I tell you that if you write a proposal saying we will investigate 1 lakh patients in a period of 2 years for 100 blood tests. I am on ICMR review panel also. Do you think that paper will get approved? Because huge sample size, 1 lakh people. Yes or no? Why? Because not feasible. I mean, 1 lakh people and so many blood tests, we don't have any idea what we are trying to do and all. Because nobody will approve that sort of research. Yours should be feasible. So grants are aplenty, provided you know where to look for them. If you are a beginner, go to the website. Dr. Rao already mentioned. So he did not put Usmania there. I am from Usmania. Usmania also has funds. We have now ICMR units in all the government medical colleges. That's called multidisciplinary research units. So our postgraduates apply to those units and the ICMR is giving fund if they approve that research question. Similar units are there in private, public teaching hospitals. So collaboration is important. We learn from each other. We give the clinical material, the laboratory gives us the lab support. So I think that's the way to go. Now, Dr. Aarti, so we've done research. And now how to publish it? Because we have heard, if you don't publish, you perish. So how do you go about publication?
So as I mentioned before, that once you are thorough with your literature research and you identify the problem, you have to figure out what is unknown, what has been already done, and what is your problem going to address. Unless and until there is no novelty factor, there will be a challenge in publishing the findings. So that's what is important, that once you do the literature research, you have to figure out what is known and what needs to be done further to enhance the study, or what is the gap there. So it is very important to figure out the gap. Then when you, irrespective of what is the impact factor of the journal, that is not important. Important is that what was not known so far and what your study is going to address. I also tell this to clinicians that whenever they are investigating any case, again pay attention to what are the unknown findings you are observing. Number of times in CKD patients, let us say you are observing chronic kidney disease patients or diabetic patients, there can be some unknown side effects of medications. There can be also some unknown findings which was not reported before. That can be presented in the form of a case report. For preclinical, you just see what is existingly that is done and what are some of the things which needs to be done further. If that unknown is there, I don't think so there will be much issue in publishing a research paper, as long as you are very, very clear that what is the research gap you are trying to address. And focus on both positive results and negative results. As Dr. Sohaib was mentioning, they did one study for spironolactone and then they found that it is not effective. Basically, that's also a wonderful thing, that you are telling that in this particular set of patients, this particular medication is not going to work. So this also gives clinician an opportunity that whenever a particular patient comes, so this sort of medications are not going to be prescribed because it is not going to work in this. So your findings are very, very important. So for publication comes the first part. Second part, the first thing is what is unknown and what your study is going to address. Once that is clear, I don't think so publication is going to be a challenge.
So thank you. So to capture what Aarti said, choose a proper journal. So if kidney disease, we choose Indian journal if it's an Indian study. And then what happens is follow the guidelines of the journal. Another important thing is rejections will be there. Learn from the rejections. They'll write some points why they rejected your article. So improve your article and resubmit again to the same journal, to some other journal. So these things, don't just give up. That's very, very important. Reviewers will ask questions, address them properly. So rejection is a part of learning process. So I mean, that's important. Dr. Rao, yes.
So just one example. Last year somebody asked me that they wanted a paper urgently, quickly. Then what I did was I have taken IMA Telangana state members list. How many people have registered for social welfare? 3,500 people. I have Excel sheet of the rejects, and I also have Excel sheet of 350 people who have died. So I have the Excel sheet. And actually, I'm not a AA expert, but I have given that 2 Excel sheets to AA and asked AA, please make a urgent publication for how many Telangana doctors have died and what is the reduction in their life expectancy. It has given me nicely within less than a minute. Then I asked it, please make tables and also a graph. It made it. It made it. It is published in JIMA. It's the Indian Medical Association data. He is the secretary. He has given me the Excel sheet of 3,500 doctors who are registered, 350 doctors who have died, and it is published in JIMA, Journal of Indian Medical Association, February this year. So can we not use AI for good purposes?
Yes, absolutely. So AI is artificial intelligence. We all are naturally intelligent. So we should harness AI for our benefit, not always in a bad manner, but in a good manner as well. And again, Dr. Rao has done what I said, finer criteria. He did something interesting, innovative, novel, and used AI to get a paper immediately. So thank you, Dr. Rao, for those inputs.
AI should be used. There is no escape from AI, and please embrace AI. That is my message to everybody. But know what AI is doing. AI can mislead you. So with Dr. Rao's experience, okay, so with his experience, he can judge what AI is doing and what it is giving. Even though it was 1 minute, he must have spent around half a day reading that paper and reviewing it and saying what was correct, what is wrong. When he was satisfied, then he has sent it. He has not relied totally on AI, which he hasn't expanded on, but I know him very well.
So point well made. Yeah.
So just to add, actually we have to declare also, like to journals. That is more important. Okay, you have to declare completely. And apart from publication, if you are doing a technology-based research, it should be patented. Patent should be one of the major criteria. So focus on patenting also, because that will give you rewards. Okay. So publication is fine. Before publication, if it is technology, just do the patent and then publish it.
Absolutely. Point well made again. So use AI. Otherwise, we have all heard of GIGO. What is GIGO? Garbage in, garbage out. That's what will happen with AI. So use your natural intelligence to harness the power of artificial intelligence. Point well made. So now the penultimate question. What is plagiarism, Dr. Shukla?
Yeah, so I was holding that AI, declaring AI as part of plagiarism. So we all know that research is meant for learning. I also emphasized that earlier. So you have to read, you have to pick from the ideas and go through the scientific literature to gather that idea. But there is a process of going it, and there is a process of presenting it. When you're collecting ideas from somewhere else and presenting it in your paper, due credit to those sources should be given. There is no restriction on presenting others' finding, but presenting without credit, synthesizing something from others' idea without credit is plagiarism. Okay, and in the scientific field, we are very, very strong on this ethical or unethical. Plagiarism is unethical. So on ethics, we are very strong on that. because that is related to integrity. Right across, we said be true to the methods, no fudging of data. All these are integrity, issues of integrity. Imagine research being unethical. What will happen to mankind? Just think of that. So ethics is very, very important component of research, and plagiarism is one part of that. If you want to see common forms of plagiarism, then just directly taking sentences from somebody's finding and putting it in your paper without. So when you are taking something directly, put them in semicolons. Credit who, from where you have taken, reference it adequately. If you are not doing that, then it is plagiarism. You might take something and change just few words here, there, usko passive tense mein convert kar doge. No. Again, that without crediting. You can do all that, but if you are not crediting, then it is plagiarism. Any uncredited idea, you are presenting, you are synthesizing any idea and you have taken it from somewhere, please credit it. Say that this is built upon. So you might not give full credit. You are also contributing to it. So this is built upon so and so et al. Okay. So it shows that you have also contributed to it, but the original backbone came from there. Then self-plagiarism. You have used something and you are reusing it without acknowledging it. You are just trying to make brownie points. No. Acknowledge it, credit it. And finally, what sir was saying, AI plagiarism. So in today's world, we use AI extensively. Okay, younger people use it even more than us. In fact, I have known people even for small email response, they will use an AI. Okay, but when it is used for scientific purpose, please, in your methodology, mention it. There is nothing wrong. And don't use it for original ideas. For synthesis, for literature search, review, synthesis of summary, you can use AI, but in methodology, acknowledge what software, what database, how did you do it. Thank you.
Thank you very much. So he has nicely summarized what plagiarism is. So one thing is, see, students are very smart with the AI, and our students sometimes they copy it and then paste it. But remember, your teachers are smart as well. So when AI helps you to prepare your thesis, it helps the examiners to check your plagiarism. So you have software tools, you have turnitin.com and so many plagiarism checkers. So we put your thesis, copy paste there, ask the AI, is it plagiarized? And it will give you the percentage, 20%, 30%. So we mark in red and send it back. So it works both ways. So be honest, that was the message. So if you do it, we are also equipped to check it. We also use AI. So last question now to Dr. Rakesh. So what do you think are the top 10 mistakes a beginner researcher makes?
So I think all has been said. Everybody has actually told us all that needs to be told about this, that what are the common mistakes you should not make, that you should understand what is a question. First, have a question in mind before you start research. Don't do just try to copy something which is already done. So have a question in mind. Design the study, see what you are going to do, what is the sample size that is required. So if you do this, these are the things that commonly go wrong. I mean, they're listed here saying that, these are things which everybody has said. Again and again, we have highlighted the fact that we need to have these, the problem clearly identified, identify the numbers that you're going to look at, what are the methods that you're going to follow. So when you do that, I think you can avoid these common mistakes. And another thing is that over-interpreting results. So this is something which has not been spoken, so I'll just speak about that. So what should you do? I mean, you should apply this so what criteria. You have done something, you think that, oh, I've done something great. So you should apply the concept of saying that, so what? So what is going to happen if this is what you've identified? So you don't have to over-interpret. Otherwise, we think that, this is the answer to all problems and this is the only answer. This could be one of the answers. So you need to put it in the proper perspective. So you can't, you should not over-interpret results. Otherwise, you'll get a rejection from the journal that you're going to send, saying that this may not be truly what you're saying doesn't actually reflect everything, reflect the answer to the problem that you're speaking of. So I think this is another point that we have not highlighted in the previous discussion. So I would just speak about that. And the last one is about the predatory journals. Try not to go to journals which are actually going to, there are many of these journals. How to identify that? Let's ask Dr. Shukla about it. How do you identify such a journal?
So by experience, we do it. So what we do is we look at journals. First of all, we look at the impact factor, whether the journal is listed. If the journal is listed in PubMed or in Scopus, then it is a good journal. And then the next thing is then you start looking at the impact factor. What I am very skeptical of and predict, when somebody just writes to you and says, then I go back and I look at that detail. Before I even respond to them, I just go back and I investigate that journal thoroughly. If they are asking me to write something, I will first go and check what it is. And if you are getting something like that, please don't just jump into the offer and go for it. Explore that journal, and these two markers, the Scopus listing and the impact factor, these are two very good markers to identify whether it is a predatory or a non-predatory journal.
So there is APCs are also a big problem, okay? So article publication charges. So the government of India has come up with the onus. I think many people know one nation, one subscription. So you can, there are covering a lot of journals where they can pay for that. Okay, so you can, that also, they have ruled out all the predatory journals. So you can go through ONUS website and just check which are the journals listed.
Yeah, that's a very good point. So for the beginners, predatory journals, many of the youngsters use, because nowadays for any sort of promotion, you need publications. And what better method is just to pay off and then get a publication in some predatory journal. So don't do it, because as we have repeatedly told, be honest about your research if you want your fellow people to respect you, your teachers to respect you, and there are always ways for checking whether the journal is a true journal or not. So I think we have had a good discussion. I would thank Dr. Rakesh for bringing out the missing points, and one more point is there, ethics, which we did not discuss. So nowadays, any sort of research you are starting, please be certain that you have taken ethics committee approvals. You can have your institutional ethics committee, or you can go to an independent ethics committee. That letter is mandatory. And the concept that mine is only an observational study and I am not giving any drug or anything is wrong, that you don't need an ethics approval for that. You need ethics approval for any sort of study, because most of the good journals want the ethics committee approval number, everything. So that's also important. So I think with this, the panelists conclude what we had to say about this broad topic, and I'm really happy that you people have been patient to stay here till the end, and we had a good crowd. We were wondering that post-lunch research topic, nobody is going to be there. But really, thank you all for making our day. And now I would open the session for questions from the audience, if there are any. Yes, anyone? Yes, please.
How original is an original research? I'm an associate professor in a medical college. So what happens is students come, they show me their, their protocol. They say, sir, this is not there in the past 5 years of NTR UHS or KNR UHS. That is the criteria for that.
So
then what they do is they go to RG UHS, Rajiv Gandhi, they select a paper from there which may be existing for just 2 years. So they don't bother. So for them, how do we say that Because my question is coming from a deeper sense of thinking. When we do systematic reviews, we are looking at secondary data. We are not looking at primary data, we are looking at secondary data. But the point is, if there is only one paper talking about one question, how do we get sufficient data to publish a systematic review? If, let us say, 100 people are here and 100 people are talking about 100 different things, how do we do systematic review? Because we say systematic review is the topmost thing, which dictates policy.
Yes. So okay, you want to answer,
I think
please?
review comes into picture only once we have substantial research for that. So usually when we are writing a review, if somebody is also writing a review, I think for example, if I am writing a review, I should have done at least 4 to 5 years of research in that particular area, then I'll be in a position to at least write something about it. Same thing for any finding. Let us say some finding is published. I don't think so we can directly jump to the reviewing of that finding unless until there are backup findings to support what has been observed. So I think that is very, very important that there are multiple researchers involved in addressing that particular problem throughout the world. So it is not restricted. For example, if I am working on cancer or diabetes, there are other researchers who will be also working in that particular field, particular target. So I read their papers, they read my papers. So it is basically a substantial research has been done, then only we can review it. Just by doing one finding and then writing a review, I don't think so that will be a right approach.
That is what my point is, madam. If there is only one paper and we are told, don't do that, that is not unique. That idea is already taken.
No, I don't think so.
Let me add to that. So when lot of students under us do their research and thesis project, and when we also propose any new research, the first thing we look at is, are we overcoming any limitations of all the previous existing research? If there are limitations mentioned in the previous research and this research is overcoming any of those limitations, then it categorizes into an origin. Is it looking at a new context? Papers have been done, research has been done in different contexts. Is it looking at a new context? So, and is it, and then the time factor also, if something is being done long time back, we would want to revisit that. Especially in cross-sectional studies, it makes sense to revisit and understand the current prevalence or risk factor distribution, etc. So that again becomes original research. So these are some criteria that I use to encourage students and ourselves also to do original research, and I think these should be promoted. So that's why when student comes to me, I say don't come only with the question. You first do your literature review, and when you are doing your literature review, also synthesize limitations. I'm not just interested in findings. I'm also interested in findings, limitations. So if you are now your research design should overcome the limitation. If you are overcoming limitations, then you can do the research on same topic that others have done, but you overcome the limitations.
One of the problems which you are saying is that they're looking only at your institution or your university and seeing what has been done in that university. It's not that. You should look at a proper literature review of around the world what has happened and then come to that question.
Majorly what happens in medical colleges is, they are only told this is a criteria. In the past 5 years, the university should not have a paper like yours.
Yeah, so I think that's why your role as a faculty is very, very important. Your role is important. See, students are beginners, but you are their teacher. They may not be informed, but you are informed. So we have to search not the university databases, but the public databases. As you are well aware, Scopus, PubMed, all those things. So I think there your role is very important, and I have great regard for teachers anywhere because they train themselves and the students as well. Great question. So just one more thing I was just say, as sir said, see, everything is already out there. Research, what does it mean? Research. Search for something which is already there, but with a new dimension added to it, something novel. So there is no point in repeating the Me Too thing. You did, I did in my population. That also is research, but do it with some novel attributes to it, right?
Yeah, so I'll just say, so there are two approaches to solve this problem of originality. One is called as a reductionist approach, and it is a global approach. In a reductionist approach, you just tie the knots, like you link the dots. And linking the dots also generates a novelty. If you have a global approach, you generally find a new targets. For example, you are doing some transcriptomics or metagenomics in a cancer, so you will find some targets. So that is a global approach. So both can lead to originality. Okay.
Major problem is now every medical college has so many PGs. To find, let us say, 120, 130 novel topics and then get ready with the protocols in 6 months so that they send it to the university. Otherwise, their PG is delayed. So we are stuck with limitation of time, and all this has to be considered by the ethics committee. Ethics committee is only one. You have 150 topics to cover, and
So one suggestion for that would be, see, what we were doing initially were fragmented research, but now what we are doing is, suppose diabetic kidney disease. So there are so many aspects to it. So we have 15 students, we have to allot them, as you said, 3 months, 6 months. So what we do is, each one is studying some specific aspect of the same disease, and don't wait for the newer PGs to join. Have the topics ready in your hand, because when you are dealing with something, suppose now we were seeing diabetes in obese people. Now diabetes in obese people, we have a lot of data from West. Indian obesity is different. Obesity starts after BMI of 23. So we started looking at that. Then male, females are different. We started looking at that, another student. So don't do fragmented research. Don't allot 100 different topics to 100 students. Be strong. Your unit should be strong in one topic. That is one suggestion. And you can contribute millions of articles on the same topic. Be prepared before the students appear at your door. That's what we should practice.
Which medical college are you working?
I was in Malareddy
Now?
I'm actually on a sabbatical.
Okay. So in your sabbatical, you can search for new topics when you join back.
She's a diabetologist.
Okay, okay.
Can I add one thing? Just 1 minute, sir. 2 days back, I was in Nagpur. I was sitting as a judge in oral presentation, poster presentation. There were 19 oral presentation, 45 poster presentations. All presentations are bad. All their topics are bad. Okay? All are, but people are very good. They are excellent. I commented, oh, slides are excellent. But the themes and topics and titles are bad. 45 posters and 19. But I was sitting with Suryavanshi, the senior most professor of medicine from Nagpur. So great people are sitting as judges, but all of us have decided that we are very happy to see 19 oral papers and 45 posters from a small place. They are from small places, Madhya Pradesh, Maharashtra, very small places they have come. So we have greatly appreciated all those people in spite of the bad quality. But we have given first prize, second prize, third prize. Do you know who has got first prize? A person who has presented data on ultrasound examination of MASH. And we told him, sir, you should have done at least but he has done only ultrasound and he has presented fatty liver data. But we gave him first prize. Not that he is the very best person, but the way we were happy is, don't bother about the quality. Right now we want quantity. We want more and more people to come out with the papers. We want whatever, good or bad. I told you, no, in my case, you have to show yourself as different from others. So you have to have papers. Please encourage all our students to have
Yeah, so
papers, good
I
or bad.
agree. I agree with that approach. See, if I look at my first presentation, it's horrible if I look at it now. But somebody like P.V. Rao sir gave me some prize for that. That encouraged me to do better. So I think we should respect the effort. People start like that. They are struggling. They have come there, presented AI. Maybe they don't have good teachers like him. So what we should do is encourage, be light, give little bit, I think, be a little bit flexible earlier, but later on, once they get into the thing, pull them tight. That's what I'm doing with my students as well. And I'm so happy to say that they are publishing papers in international journal with good impact factor, with themselves as the first author. So I'm telling them to become first authors in their research. I'm the head. I guide them, give the idea, but I'm there as corresponding author, second author, or whatever author, but they are so excited because they are having their original research. So I think encouragement from people who are our seniors is really important. Yes, please.
Good afternoon, ma'am. So my question is on study design. So when we do a research, usually say for example, you take the example of semaglutide. We are studying only one molecule and its effect on body, right? And with placebo, randomized controlled trials, double-blinded and all. So it is very easy whether the drug is effective or not. But here we are talking about lifestyle disorders. Say for example, thyroxine. I train doctors and dietitians in lifestyle medicine, and there are hundreds of students, thousands in fact, all over the world. So they are able to bring down the TSH levels to normal by changing their lifestyle. When I say lifestyle, we talk about 7 types of nutrition. So the physical nutrition, the food we are eating is only 1/7. So there are other 6 types of nutrition. So I teach all of them to look for the risk factors in their different nutrition. There is social nutrition, vocational nutrition, psycho-emotional nutrition, and we look for those factors. And if you do the permutations and combinations, say for example, when I look back at the data that my students have come up with, at least 200 thyroid cases, now they are thyroxine free, right? So their TSH levels have come down, their energy levels improved. So if I were to present this data as a paper, I mean, how do I study the design? So it will not come under this gold standard which we call as like double-blind RCT, right? So what I encourage my students to do is like do every case as a case study. So is it the only way, or is there something else that I can do with any research institutes?
You can take that.
Yeah. So what you are describing comes in the category of complex health intervention. Okay, and there are study designs to assess complex health introduction. Dr. Nuruddin also said research are either reductionist or global. So you look at global factors or you reduce them down to one. So there are designs, we can talk about that later on.
And just to add to that, when you're looking at a global this thing, when you're looking at many factors, then you have to have a study, I mean population has to be much bigger. You'll have to have very big population or sample size to come to saying that, okay, this was, whatever you did was important. Or if you reduce it down to, a single factor, you want to say a particular molecule also you're studying, just one factor, or a particular intervention you're studying, particular lifestyle intervention you're studying, then you can have a smaller sample size. But so this is important, that you should decide what you are going to study, what you want to study in a particular study. What is the aspect that you're going to study? Be sure about that. Not giving a very broad.
Yes, sir. I get your point to say.
Whenever you are doing any kind of clinical study, for example, there will be always a primary question. So your study is going to revolve around the primary question. Then there will be a set of secondary or subsidiary questions which will help in answering the primary question. But you are basically, one factor which will be the dominant one compared to the others. Although you are saying you are to pick up the 7 factors. But then the primary, what was the primary factor which you were trying to address? And then others will revolve around that or correlate to that primary factor. So I think one major factor has to be there, and then other subsidiary or secondary factors will be then trying to correlate with the primary.
Yeah, so thank you for that question. And as I understood that you are trying to look at the thyroid levels with different diets, some 7-8 diets. So, and as I understood, you are looking at 37 factors and 100 people or whatever. So keep the biostatistician on board from the very beginning, because 37 factors, 100 sample size will not be adequate. So what you have to do is take a much larger sample size.
I just gave you throw the numbers.
Yes, I'm just giving an example. I got your question.
Yeah.
So see, any study can be addressed, and statistics is a beautiful tool. Statistics has its own drawbacks, right? But the thing is, we should use it rightly, because otherwise we'll just end up saying maybe this is better or maybe that is better. Statistics gives a objective answer. answer which in numbers we can compare and understand. So get the statistician on board. For every type of question, there are different types of studies. There may be an observational study, that may be a case control, that may be a cohort, and when you are comparing a randomized controlled trial. So I think you can discuss one-to-one with a biostatistician with your problem.
Take it up little later because we have to conclude. We have to finish.
I think
Thank you.
yes.
Thank
yes.
you.
Running behind time.
Fine. So I thank all of you for the massive interest shown, and I thank the Idea Clinics for the great opportunity.
And
thank
all the
you.
panelists for the time.
Yeah, and yes, absolutely. And all the panelists, thanks. That's my better half speaking, right? So thank you very much.
Thank you so much for such a productive session.